Imagine having a supercomputer at your fingertips. What will you do? This is what the CEO of NVIDIA, Jensen Huang, said, that every individual will have a AI personal assistants, not only in the sector of computer science, but also in biological simulations, drug designing sector, protein folding, etc. So take an example of this meta glasses. You can take a photo, you can record the videos, you can ask the meta AI that you want to know. For instance, you can take a picture like this by click of a button, and you can record the videos as well. So it's like an AI personal assistant. So imagine that situation in terms of drug designing sector, how it will revolutionize the drug designing, industry. Billions of dollars, billions of euros that has been invested in the sector of drug designing will be reduced to a lesser amount. So that will revolutionize. So NVIDIA launches a generative AI model for bioinformatics, I would say for drug designing sector, and also for molecular modeling. Today I'm going to show you how you can also do molecular docking by using NVIDIA supercomputer online. You don't have to download any kind of software, or you don't need any kind of resources, but you can perform the molecular docking for millions of molecules at your fingertips. So check out this page where NVIDIA launches DGX Cloud, giving every enterprise and also to every individual to access the supercomputer. So if you go to this website called NVIDIA, and you can find, if you go back, so you find different types of models over here. This is Llama, and this is Mistral, this is Llama again. But if you go to the biology section, you can see AlphaFold 2, DiffDock, MolMIM, and also RF diffusion, protein, and also ESM fold as well. So today we are going to explore this DiffDock because we'll be dealing with the molecular docking analysis. So if you click here, so you can see the first page is all about the example that they have shown. So this example is this particular drug and the MPRO of SARS-CoV-2. So they have already docked it, and you can see different types of ranks and poses, etc. Over here. Okay, but we are going to upload a new molecule and a new protein file to check how much time it takes and how you can perform the molecular docking analysis by using DiffDock of NVIDIA. So I'll be dealing with Remdesivir. This is a popular drug back in 2020 during the pandemic. So I downloaded the SDF coordinates. So it accepts only the SDF file. And then you go to PDB and download the structure of your interest. So I'm downloading this MPRO, and I pre-process it in Chimera X by removing all the ions, all the water molecules, all the ligands associated with it. So I have already downloaded these two molecules, and now if you go to this DiffDock and upload a new molecule, that is my drug, and then the PDB file, which is mpro.pdb, and number of poses I keep it to default, that is 20, and diffusion steps I keep the default at the moment, because when you read the paper then you will get to understand what these steps means. So I keep it to default and then click run. So now it is generating all the poses, and within a few seconds it will show all the results, and you can investigate those results as well. So now it's done. You can see 21 poses to the binding site, and you don't have to define anything, not any sort of grid box or any sort of active site molecules, active site residues, etc. So it can do your analysis automatically, and you can see all the poses. You just click here, then you have this rank 1. This is your rank 1. You can also zoom in. Yes, you can also zoom in, and you can see all the ranks. Okay, this is rank 1, rank 2, rank 3, rank 4, etc. Okay, and then you can download all the files. So I just click download, it will download all the files. And also one more thing that I want to show, that they have not only this graphical user interface for molecular docking, but they also have provided the Python code, but you need to obtain the API key. This API key you need to register, and they will provide you, I think, a trial version of this API key, and you can use that API key to perform your analysis, a virtual screening for millions of molecules. Okay, this is Node.js, this is shell script, okay, and also this is Docker container also they have provided. You just have to paste your API key over here by registering. So if you click this, so you get the free API credits. You just enter your email ID and you will get your API credits. Okay, so yeah, so let's investigate what they generate. So if you go here, they have this folder DiffDock. So if you click here, this is your pose confidence. This is rank 1, rank 2. So how they rank? So -1 is the highest rank, -4 is the least rank. But in general, when you perform molecular docking, so -4 should be your highly ranked molecule, right? But in this case, it's kind of different. So to understand that, you need to understand the paper that they provided over here. This is the paper. You can read about how the algorithm works, and this is the DiffDock GitHub repository, and you can see this is your ligand and protein, this is DiffDock, and this is the ranked poses and confidence score. Okay, I'm not going into the details of the paper, but you can read the paper and you can understand how the confidence scoring, how the scoring patterns they have calculated for this. So this is a generative AI model for molecular docking. You can consider this as one of a kind AI model for your molecular docking in comparison to the conventional tools that you use for Autodock or Autodock Vina. So this is kind of good for your project, and also you can showcase this project for your master thesis or bachelor thesis, etc. Okay, but we'll investigate this one. So we load the target protein, we open it with Chimera X. Okay, this is your structure, and then we close this. This is your structure, and then we load one by one. So this is your first molecule that has been docked. You can select the residue, molecular display, and then H bonds. So you can actually see how it has been docked. You can see the poses as well, and you investigate whether these hydrogen bonds make sense or this binding pose makes sense or not. Okay, so clear it out, then select all and action focus, so it will recenter it. And now they also provided this reverse process. I mean, how the ligand binds to that particular position, they also provide that process as well. So you can read the paper and understand how this reverse process works. So if you just drag and drop here, you can see all different kinds of, I mean, this is like a finding the best possible binding site for your protein, or finding the best possible active site for your protein. So this is kind of a reverse process to that. If we read the paper, then we might be able to understand how this process works. But for your thesis, I think you don't need to provide the details about the reverse process, but rather the binding poses. But if you are interested, then definitely you can have a look into the paper. So yeah, this is how they're done, and these are your 21 poses, or 20 poses, I guess. Yes, 20 poses, and you can perform all the analysis. And check out the GitHub page for DiffDock, and they have provided how this analysis has been performed. So you can see this is the reverse process. So it binds to different areas and find a suitable area to actually bind the molecule to that particular active site. So this is your confidence score. This is your second confidence score.
yeah, it's kind of cool to have this. And also they have provided this example. So if you go here and if you go to the projects, they have this example projects. So try this project, and they have some notebooks, also Docker file, so you can open this and try out yourself. You just have to paste your API key here, and the rest of the process will be done automatically, I think. Yeah, this is a cool thing to showcase your projects. You can also try out all the other different tools. This is for molecular docking, but also you can try out for protein AlphaFold 2 and protein docking as well. This is kind of popular. So generally we use Class Pro 2.0 for protein docking, but now this RF diffusion has is proving to be very good for protein docking as well. So you can check out all the analysis, and you can present your thesis as a master thesis or your bachelor thesis for your project. Yeah, so that's it for today. Thank you very much, and I'll see you in the next video.
If you like my contents, then please do subscribe to my channel. You can find out all the drug designing videos over here and the molecular dynamics simulation videos over here. So check out all the tutorials and please subscribe to my channel. Thank you very much.